nueva temporada de los seminarios del Laboratorio de Ecología de la Conducta de Artrópodos, en este ciclo que le hemos titulado Ecología de Insectos en un Mundo Cambiante. Para todos los que ya nos conocen, pues qué bueno que están ahí de nuevo. Vamos a seguir así con estos seminarios que han resultado muy interesantes para mucha gente. Y es un placer para mí presentar a Idelle Cooper. Ella es una profesora asociada de la Universidad James Madison en Virginia. Ella hizo su doctorado en Indiana University y luego hizo postdocs en la Universidad de Wisconsin y Michigan. Y trabaja esencialmente la variación en color y el dimorfismo sexual en libélulas y sobre todo libélulas de las islas hawaianas. Ha tenido apoyos importantes de la National Science Foundation y está cercana a nuestro corazón porque ella ha estado colaborando, hemos estado trabajando con varios asuntos de coloración recientemente. Entonces estamos muy contentos que ahora a ella le toca estar del lado de los ponentes. So thank you very much for being there. To all of us, to all of you that are new, welcome. Those of you that were present in the past and last year, welcome back. This is the first seminar in this season, and we have a very nice speaker today, Idelle Cooper. Idelle is an associate professor at James Madison University in Virginia, and she got her PhD from Indiana University, and then did a couple of postdocs at universities of Wisconsin and Michigan. She's now very much interested in color, in insect color, and particularly in odonates, in damselflies in particular. She has done lots of work, very fascinating work actually, with the color variation and sexual dimorphism in Hawaiian damselflies. For all of you who are interested in color, you're gonna, for sure, you need to read her papers. They're really nice. And it's a pleasure also to have her here because she is a collaborator. We have been working together along with Kata. She's possibly around there. And we are writing this review, which is taking forever, but anyway, on color in dragonflies and damselflies. So thank you very much, and we're gonna hear Idelle now. Thank you, Idelle. And yeah, if any of you have a question, please you can either type it, write it down on the chat section of this Meet program, or you can wait until the end and ask her directly. So thank you very much, Idelle. Please go ahead.
Thank you so much, Alex. Thank you so much for inviting me, and it's so nice to be here with you virtually, though I really hope to be there in person sometime so I can chat with you and Kata in person and catch damselflies, obviously in person. So I'm perhaps foolishly presenting this on a new program for me. So I can't see my own video. I don't know if you can see it. And I can't see the chat thing. So if you want to ask a question during it, or Alex, if you see a comment, just speak up and that's fine. Otherwise, I'll just come back out of this display at the end and then we can talk more. So I'm going to be talking about sexual selection and species recognition in jewelwing damselflies. And this is a new project for me, fairly new. So as Alex mentioned, I've mostly been studying color variation in Hawaiian damselflies for the last, 18 years. So, but to find something closer to home that I can have more students involved in, I've been starting some projects on these jewelwing damselflies, which as you can see have this dark wing and interesting wing variation, which are really lovely. So one of the main questions in nature and evolutionary biology is what causes color variation as something that we see so obviously in nature. And so on the left here, upper left, you can see these damselflies, these are the Hawaiian ones that I've been studying for a long time, in which I study their sexual dimorphism in particular, in which the males are red and the females are green. And you can see this in this mating pair of damselflies. So I find that really intriguing, and I'm not going to talk about that at all in this talk, but it's related because it's still trying to figure out how sexual dimorphism evolves. You can see sexual dimorphism in all sorts of other animals and plants. You can see here on the upper right, these elephant seals in which males are much, much bigger than females. That's not an infant, that's a female here. And then on the bottom left, elk, in which males have antlers. Lots of other organisms have battling structures that are sexually dimorphic. Plants also are sexually dimorphic, as you can see here. Just one example. And then peacocks being a classic example of sexual dimorphism, in which the males have this amazing display that females don't. So damselflies, I think, are particularly nice study organisms to look at these kinds of questions because they're abundant and they're easy to catch, and they have some really dramatic phenotypic variation that we can study. So on the left here, the Hawaiian damselflies again, and today I'm going to introduce my ideas in Caloptrix damselflies, which are also sexually dimorphic. In body color, somewhat, though it seems like the wing pigmentation is even more dramatic in sexual dimorphism. Before I get started on all the studies, I want to first acknowledge the many, many students who have worked on these projects. So this is just the students who have worked on the Caloptrix stuff, not the Hawaiian stuff. Suzanne being a master's student of mine, and Melissa. And then the rest are undergraduate students. And as much as I can when there's not a pandemic raging, I take students out to the stream so they can also catch the damselflies and measure aspects of their phenotypic variation. So as we know, sexual selection often leads to these patterns of sexual dimorphism. And this happens through the selection pressures of male-male competition, in which males are competing for access to females, or territorial sites, and thereby access to females, and also female choice of mates. So this is the general idea about how sexual dimorphism evolves, though my work in the Hawaiian damselflies indicates that sexual dimorphism isn't always due to sexual selection, that other kinds of selection, ecological selection that doesn't have to do with mating, can also cause this difference between males and females if males and females are under different selection pressures ecologically. Here are some pictures of some Caloptrix species, splendens on the left, and then aquabilis in the middle, and maculata on the right, all of which have different levels of sexual dimorphism, especially in their wing pigmentation. So how does this sexual selection pressure come about? It's often due to the fact that female investment is often larger than male investment. This is not always the case, but this is generally the case, it seems. Female eggs are larger and tastier than male sperm, and females have a larger investment on average than males in reproduction. So females have few eggs, and so each of their mating opportunities has a larger effect on their fitness than male mating opportunities, because males have a lot of sperm, a lot more sperm than they have potential mates. So this results in males competing for access to mates because they're limited by the number of mates, while females are limited by the number of eggs, and therefore there's selection for females to be choosy of male mates and potentially select on the highest quality male, not just as many males as possible. So this is the basis of differing investment of males and females in general. So I want to show you some of the behaviors of these damselflies, these Caloptrix damselflies. This is Caloptrix maculata. The common name is ebony jewelwing, which isn't that a lovely name? They're like little jewels flying around the stream. And so you can see in their mating behavior some indications that they're under sexual selection pressure. Males for competition for females in territorial sites, and females may be choosy of males. So you can see in this video a male guarding a female. So that's the male flying around. The female is actually going underwater to oviposit her eggs. So she puts her eggs in plants underwater. Other males are flying around and other females are flying around. It's a circus of mating on the stream. And so her mate is chasing away other males who are still trying to get to her and grab her and begin copulation as well. So you can see there's a lot of behaviors going on that display. Males are displaying to each other as well. So males display to each other to hold down territories, and they also display to females potentially as for females to choose them as their mate. So while there's lots of evidence of sexual selection, multiple selection pressures can act simultaneously, which is important to remember. And even within the classic examples of sexual selection leading to sexual dimorphism, like in the peacock tail feathers, at some point, sexual selection might drive a trait to the point that it's under stronger natural selection or other ecological selection. So you can imagine how male peacocks, they're selected to have these large and elaborate tails, but at some point they can't fly, run away from predators, or get food, or even mate. So there's some balancing selection eventually from natural selection to stop the directional sexual selection on that extreme trait. This seems to happen more strongly in females. So females are typically under less strong sexual selection than males in many species that have been studied. And this can lead to a relatively strong ecological selection pressure on females relative to sexual selection pressure. We can see this in some butterfly species. So here on the right is a species, Hyptolimnus. The male is at the top here, and there are 3 morphs of females along the left side here. These female morphs have been selected to mimic a closely related Danaus species of butterfly that is toxic. And Danaus is in the same genus as monarchs, which are also toxic. So because females are not under such strong sexual selection, that natural selection drives them to have this mimicry to prevent getting eaten, while males are under stronger sexual selection and therefore they don't have that mimicry. So multiple selection pressures can act simultaneously. However, these different selection pressures may conflict with each other. So 2 of the roles of traits that I'm interested in particular that I think conflict in the literature, they conflict, in theory they conflict, and that is species recognition. So there's selection for an individual to choose a mate that is conspecific. Additionally, there's selection, as we just talked about, sexual selection, which is preference for a particular mate given a choice. So these are both elements of mate choice, yet they might have different goals and drive traits in opposite direction, even the same traits in opposite direction. So this has been described in a theoretical framework by Mike Ryan and Stan Rand in a 92 paper. And so let me show you this model in a figure. So you have a male trait along and the x-axis, it can be something like male wing pigmentation, for example. And then, so species A will have a normal distribution of that male trait. And you can see on the right here in blue, females, the female preference for that male trait in sexual selection. So sometimes the females like average males. When that happens, that one trait, whatever that male trait is, like wing pigmentation, can signal both species identity and mate quality, because females are going for the average of the species. But there's a lot of evidence that sexual selection is actually often happening for more extreme male traits. There's usually directional sexual selection for more extreme traits. And there's a lot of evidence in animals and plants for this. So that's all and well, except what if there's a second closely related species in the area that have that more extreme male trait? So that females from species A, because they prefer males that are brighter colored, louder calls, more elaborate displays, prefer instead, when given a choice, a different species. So in those cases, sexual selection may conflict with species recognition. There are different goals of each decision. Sexual selection may select for males that have the highest quality and maybe the most extreme traits that indicate that quality, while species recognition doesn't want to make any mistakes. I mean, I say that in as though it's thinking, which it's not. But there's selection in species recognition to be quite conservative and safely choose the conspecific mates. Here's an example of such a conflicting process. So this example is in spadefoot toads, a Phoenix study in 2000. So this toad on the left is Spea multiplicata, and there's variation in male call rate, which you can see in white here. Females of that species prefer males with a higher call rate, and that seems to indicate some male quality. In areas where they're sympatric, where they live together with another spadefoot toad species, Spea bombifrons, females have been selected to adjust their mate preference so that there's a compromise on male quality to ensure conspecific matings. So it's in these sympatric areas that females no longer prefer males with the more extreme call rate, even though they might be the best males, because they might indirectly or unintentionally choose different species. Often when there's heterospecific matings, there's some fitness cost to the offspring. So there's often selection against mating with a different species. So this conflict happens in nature. What resolves this conflict? So as Ryan and Rand in '93 describe, there can be different male traits that are being selected on. And so I show this in this graphical way, showing a three-dimensional graph of male trait along one axis here, and the female preference for that trait for sexual selection to choose the highest quality male, and then an independent, a separate male trait that is used by females to select the conspecific males. So in this way, these two male traits can evolve independently, and that sexual selection and species recognition don't conflict and push that same trait in different directions, and thereby stop response to selection. There's some evidence of this happening in nature. One example is in pygmy swordtails. by a paper by Hankinson and Morris in 2003. And in this species, there's selection by females, sexual selection by females for larger male size, which indicates male quality, but they first choose conspecific males based on male chemical cues. So there are two different male traits that are under selection for these two different functions. So here's my alternative hypothesis. I think that there can be two traits that evolve, but that gets a little complicated because then two traits need to be under selection with variation in those traits and selection by various signaling modalities and detection of that specific signal for those functions. Obviously it happens, but something that might be a simpler evolutionary pathway is this, that a sexually dimorphic trait could be used for both of these functions, sexual selection and species recognition. So there are two axes now, but they're not two different traits. They're just one trait, and the difference is that they're in males and females. So that one trait, like wing pigmentation, can communicate both species identity in females and male quality in males. So we have lots of examples in nature of sexual dimorphism evolving in one species, in one genome, but males and females are being selected to go in different directions on that one trait. So this is the hypothesis that I want to investigate in my upcoming research. So in this way, there can be directional sexual selection, as you can see along this axis for female preference function. And then more conservative, stabilizing selection of species recognition, you can see on this axis, in which females, it's on the female trait in this case, that males select females to court, for example, who are conspecifics. So I started these studies using these two Calopteryx species, Calopteryx maculata, the ebony jewelwing, and Calopteryx aquabilis, the river jewelwing. So I'm going to describe some preliminary studies that I and my students have been doing. None of this has been published yet, but it's the basis for what I hope to be my next grant proposal and next 5 years of research or so. So what you can see in this photo, first off, is that there's sexual dimorphism. So I show these, here on the left is the maculata with fairly dark winged females. Aquabilis, however, on the right here, can have females that are either dark winged, similar to maculata, or light winged, which look quite different from maculata. And this all depends, it seems, on whether those species are in the same area together or not. So I'll describe that in a moment. So I've been collecting over the last many years these damselflies and all of these populations, these dots here, throughout the Midwest and into Canada. So here's the species ranges of these two different species. Calopteryx aquabilis is more northern, so it's in Canada here where it lives allopatrically or by itself. And then the southern edge of the range is this line, this dotted line along the bottom here. So it goes into the United States, into Michigan, Wisconsin, Minnesota, and the top half of Iowa. Calopteryx maculata is a more southern species. So it's down here in the southern part of the US, it goes quite far south. And it's the northern border of its range is up here in Ontario, just at the top part of Lake Superior. So this is a fantastic variation in where these populations are, because you can get to allopatric populations of aquabilis in the north, allopatric populations of maculata in the south, and sympatric populations along this middle area where both species exist. And what's been documented a long time, about 40 years, is female wing pigmentation that differs according to whether these populations are allopatric or sympatric. So I just show you the wing pigmentation of the females here on the right. Aquabilis have females with dark wings in the north by themselves, which is very similar to maculata wings. When you see them out on the stream, it's really hard to tell the difference. But in sympatric populations, aquabilis females are much lighter, and maculata females are about the same. They're slightly darker. So there appears to be character displacement, which is selection that drives this trait in females in different directions in sympatry. So the video that I showed you before showed a female going underwater to oviposit. Who knows why that is? It probably has to do with avoiding some mating harassment by males. But I have this video to show you because I think it's fascinating that these flying insects are underwater to oviposit. I just put the camera underwater and see she's putting her eggs in that piece of grass. And then she just lets go, floats up, and then pops out of the water. So I think this behavior, though, indicates that there's there's a lot of mating activity that's exerting some strong pressure. So in these sympatric populations, I think there's a lot of interesting things going on. And at first, when I heard about this, I was like, well, are they really in the same area? These two different species, are they, even though geographically they're in the same area, are they in the same microhabitats? And they are indeed. So here's some photos showing that. So this is in that middle range of their species distributions. This is a maculata male on the right, an aquabilis female, just perched on a stick right next to each other. Aquabilis female, all in the same plant. And then here's a photograph of an aquabilis male putting his wings out in a courting display to this female maculata who's ovipositing and being guarded not very effectively by her mate, which is a maculata male. So because in these sympatric populations, these damselflies are in the same area, there's this possibility for sexual selection and species recognition to be happening at the same time. How do they conflict? And if they don't conflict, how is that resolved? So you can see in this video, there's an aquabilis male up here on the stick perching and a bunch of maculata females ovipositing and maculata males flying around. This is all happening in the same space. So this becomes additionally interesting because males have the possibility to scrape out the sperm of previous females. So this is a classic and somewhat disturbing, if you think about hard, study of Jonathan Waage in 1979 in Science called Dual Function of the Damselfly Penis, Sperm Removal and Transfer. And it has a scanning electron micrograph images of the male penis. So here's male penis on the left, and then after scraping out the sperm of previous mates from a female, you can see in this middle section, this middle photograph, sperm on these hairs that are going backwards. And it also has these little horn processes that can scrape out the sperm from sperm storage organs that females have adjacent to where they have their eggs. So females can store eggs, but the males can scrape them out with these horn processes. Here's a close-up of that horn process on the right here with hairs that are going backwards and sperm, this is the SM here, that the hairs are grabbing sperm and scraping out the females, which means that this kind of sexual selection can happen. The males are fighting each other for territories, but they're also continually fighting for access to females because the last male to mate with her before she oviposits is the one who fertilizes her eggs. So in this species range, these two species ranges, we can conduct these studies of what is under sexual selection, what traits are under sexual selection, and what traits are under species recognition. And we can have some hypotheses based on what traits are varying, and it's mostly varying in wing pigmentation, especially female wing pigmentation in sympatry. So this is just to remind you, in allopatry, the wings are dark, and then in sympatry, the aquabilis wings are light. So Waage in '79 measured this displaced wing pigmentation of females, which you can see here on this figure, in which the y-axis is the region that corresponds with the map on the right, so that in allopatric region of aquabilis and maculata, they're both dark, as you can see, but it was actually measured. And then that trait diverges in sympatry, as you can see here, with aquabilis females getting lighter and maculata females remaining dark. So that's what Waage showed in '79. When I started studying it, I measured the same thing to see if it was still there, and also measured male wings. So these are just female wings here. So here are my measurements. So it's the same region along the y-axis. So allopatric aquabilis and allopatric maculata, dark wing females, and then this displaced character in sympatry, as we already knew. And then males, they don't really change so much between allopatry and sympatry for both of these species. They're already pretty different. So there seems to be selection on the female wings in particular to diverge in sympatry. Interestingly, I also went to Utah, where there's an allopatric population of aquabilis that's really far from this Ontario, Canada, those populations, and they looked different. The female wings were lighter, which are more similar to the wings for females in sympatry here, which is curious because they're by themselves there. They're allopatric in Utah. And I think maybe it's because it might be a similar selection pressure to what's happening in Claptrix splendens in Sweden. So this is a study by Svensson and Freiberg in 2007, in which they show that there was selection for lighter wings due to predation by these wagtail birds. And this is a fantastic study because these birds grab the damselflies and go to a perch and then rip off their wings and slurp down the bodies. So unlike most predation in nature, you can actually see the trait of those who are eaten. So there's selection to be lighter colored because of bird predation. And when I was collecting in Utah, in these streams cutting through some fields, there were birds everywhere. And even the damselflies behaved, they were flying in a way that was really jittery and erratic in a zigzag form, which indicated that they might be under selection pressure from birds. That's an aside. So coming back to the main study though. So once again, here's my hypothesis. It's a sexually dimorphic trait that's being used both in sexual selection and species recognition. So I'm trying to figure out what are the best studies to do to answer these questions. And I have some preliminary studies trying to figure out what works and what doesn't work. So I'm presenting to you today in large part to try to clarify my own thinking and to get your feedback about how to address this in the most comprehensive and effective way. So we can use this guide, this figure, as a guide to the different studies that are necessary and the predictions that we can have. So here are the questions. Does wing pigmentation function in sexual selection on male wings and species recognition on female wings? So for this first one, does it function in sexual selection on male wings? We can take this part of the theoretical figure and see whether this is true. Is there female selection for more extreme male traits? And the prediction is that there is two things, I think. I think there is sexual selection going on male wings compared to female wings. And one way that you can see this is whether there's greater variation in mating success in males than in females. So are males exerting, is there selection pressure because of this greater variation in male mating success? That is an indication of stronger sexual selection. And also, I haven't gotten to those data. I haven't gotten data for that yet, but do males with darker wings have greater mating success? Is there directional selection? And that I have some data on. So here's how I started studying this. I did an experiment just in the stream in the allopatric population of maculata. This was in Michigan. So I started off with just a study of 1 week and 400 meters of stream in which I collected all the females every day and marked them with a fluorescent powder. So I dipped the females in powder and then all the males every day to see which of the males had powder on his penis that he got from the females. So if he mated with one of the marked females, granted, I couldn't mark all the females, but I marked thousands. Those marked females will transfer the powder to males. Instantly, I repeated this for a 1 month period and then also marked males to see which females mated. So I also marked individuals with unique identification paint combinations. So you can kind of see it here on the underside of this female here. There's a series of dots which indicates like she's number 172, for example. And you can see it a little bit here. It's fuzzy though on the male. So I know which males and which females are which and could scan them. So using a computer scanner that's battery powered, which is fantastic. You can take it into the field, very gently put them onto the scanner, hold them down with some glass slides, scan them, let them go again, but then you know what they look like and whether they mated, and you can let them go to mate again, potentially. So in this first study, I scanned males to measure wing pigmentation and see whether it differs between males who mated and who didn't mate. So here are the data that I have of, so here's 4-wing pigmented area of males and females. And so the dark circles indicate unmated and the light circles indicate mated for males and females. There was a difference in that pigmented area for males, not for females. It was the darker males who were mated. And I also measured, which I forgot to mention, I also measured where the male, I took note of where the males were along the stream and whether they were repeatedly in the same area, which indicated male territoriality. So you can see in this table of an ANOVA analysis, the wings differed depending on whether males were territorial and how dark their wings were. Mated males are territorial and have larger signals. This is in agreement with other studies, also in Calopteryx and Calopterigidae damselflies, including Heterina americana. So you can see this study, including Alex here in 2008, all indicating that there's sexual selection of females on male wing pigmentation. So it does look like that in Calopteryx maculata, at least, I'm not sure about Calopteryx aquabilis, I haven't done that study yet. There's directional sexual selection of females on this male wing pigmentation. So on this axis, on this x-axis. So that brings us to the next question. which is, does wing pigmentation function in species recognition on female wings? So that would be this axis here. So is this true? The prediction is that female wings are under stronger selection in sympatry than allopatry because they're two different species. And so there'd be stronger species recognition selection. And so we expect divergent selection between the species, character displacement, and we expect females of the rare species to be under stronger selection from males of the common species. So in trying to figure out the ideal study, I'm going to go through, like, hypothetically all the things that I would like to do, but let me just show you what I've started out with so far. So doing these tests in which we have females from allopatry and sympatry, and they could either be heterospecifics. That tests the effects of all heterospecific traits if we just move females around and see whether males mate with them. And we could do this for conspecifics too. And the reason to have those females is really a control for the experimental setup. So I started off by doing this in these 3 populations in which I got allopatric females from Canada and southern Michigan, and this is a little wonky. This should be over a little bit, and not in the lake. But so this population is right here. So in this sympatric population is where I did these studies. Here's a picture of my parents helping me collect endearingly. I collected females from allopatric populations in these cages, got permits to take them across the international border and put them in coolers to keep them cool. Here's some people who have helped me in undergrad, Hannah Caffey and Tom Getty at Michigan State. So what we did was put them in tents and then dip them in the powder, see who mated with whom. It worked great half the time. So the maculata males mated when in the tents, but for some reason, despite a lot of effort, the aquabilis males would not mate in the tents. I don't know why. We tried to move them right next to the stream, keep them happy. That's something I don't know the answer to. So I'll just show you what male mates, what the maculata male selection was about. So here are the initial tent experiments with maculata males from sympatry and whether they formed tandems, which meant that they grabbed the females to initiate copulation. So you can see the powder on the females next if that happened. So, so here are wings from allopatry or sympatry for females that are either heterospecifics, so they're aquabilis females, or maculata females here. And you would predict that these males from sympatry would prefer the dark-winged of the other species. If they're willing to mate with the other species, we'd expect it would be the dark-winged ones because it looks like their own species, their own females. And indeed, that's what I found. So here's the number of females who are mated over the total from these maculata males. And so using a Fisher's exact test, there's males preferred the dark-winged heterospecifics to the light-winged ones of their own species. Granted, it's not a very large sample size for the number of who are actually mated in sympatry. So there's a significant difference in the heterospecific females, but they didn't really have a preference for conspecific females as predicted. So that worked well in the tents. But let's think of all of the different permutations of what we can do. So I just talked about these two. We can also, ideally, what you'd want to do is to switch the wings around because it's really the wings that are the trait that I think are under selection. So we should keep everything constant and change the wings. Ideally, it would be something like this, where you have heterospecific with conspecific wings, conspecific damselfly females with heterospecific wings, and conspecific with conspecific wings as a control if you were to do an operation to move wings around. And again, you can do this with wings from allopatry or sympatry, so you have light and dark wings. So we collected damselflies throughout this range and did this. Largely, it was the work of my master's student, Melissa Encenius, who really perfected the art of cutting off flying insect wings, gluing on other wings in a way that they could still fly around. So let me show you this video that she made. So this is under a dissecting scope, cutting off the wings. In this case, just to show you, she's gluing on a male wing. So leaving a little stub and then using this, after trying all the glue we could ever find, glue that was used for fly fishing flies, that was UV curing, instantly dried and was lightweight. And so these damselfly females could fly around, not forever, but for a couple of days without their wings falling off. So they could fly around and mate. This gives us great potential in doing all sorts of manipulative experiments on the wing pigmentation specifically. So I'm going to show you briefly some of her data, in which this again is in tents, only with maculata males, because they were the only ones who would mate in the tents, and lots of combinations of females from sympatric and allopatric populations, in which you can see the body type here and then the wing type in this column. And then just to simplify things, just whether these wings were dark or light, you can see here. And unlike what I did before, which was to see which of the females were mated with, she individually marked male with different colored powder so she could see male preference, which I think is a better way to do it. So somewhat small sample size, so she combined different treatments to reveal how males, the maculata males in sympatry, prefer their conspecific females if they have dark wings, but not if they have light wings. Similarly, when there's the heterospecific females, they prefer also dark wings. So in all cases, these males who have dark winged females prefer dark wings in sympatry. So we're trying to figure out at the end of our last field season, what can we do to test this more thoroughly? Because right now, up to that point, we could only test it in tents, only with maculata. How do we test it with aquabilis? Maybe they mate in the stream, so maybe we can just bring the damselflies to the stream. So we started some trials of tethering females with really thin fishing line. It's actually a common way to do behavioral studies in damselflies, as strange as that sounds. And then setting up this camera to record male mating behavior toward those females. And you have two different options of females near each other. So two females were tethered. They were given a little perch of a lily pad here. And that worked quite well. So let me show you this video that Melissa took. So there's an aquabilis male. He's readily mating with one of the two females. There's a female here and then a female up on the other lily pad. We can also just reach in, grab that male and put him in a cage. So he's made his choice. Other males are around though. Here's another male, also selecting that same female, adamantly. And then we could grab him too and then see what other males select which female. So here are the predictions, all based on what are the males seeing of their own conspecifics in those populations. And I won't go through it all because it gets a little confusing, a little complicated, but here are the main comparisons that we expect there to be a difference. So we expect there to be a difference between aquabilis males who are sympatric versus allopatric. So sympatric males, we expect them to prefer light wings compared to allopatric males that prefer dark wings. We also expect a difference between maculata males from sympatry and aquabilis males from sympatry. That is the character displacement that we're expecting, that maculata males will select for dark-winged females to approach and court, and aquabilis males will select for light-winged females. This is the displacement that we're expecting because of species recognition in sympatry. So here's some initial data that Melissa got. So let me just summarize for you what it shows. These are the number of males responding in those tethered experiments out on the stream. And it's given in males that show a preference for dark-winged first and then light-winged second as that second number. So it's just simplified into dark and light wings. And for now, removing which species the wings came from. So males, as predicted, aquabilis males preferred darker winged females in allopatry, and even though it's not a large sample size, the opposite sympatric females with light wings. So just like over here on the right. And then in sympatric populations, aquabilis males who prefer the light wings, that differs here. That differs from maculata males that prefer dark wings, as predicted. So we need to continue this for a larger sample size, but there's some indication that there is this character displacement selecting for different female wing pigmentation in the process of species recognition on female wings. So the reason why we think that it's the aquabilis females that change the most between allopatric and sympatric populations is that Melissa recorded the abundance of the different species in the sympatric populations that we studied. And maculata males are much more abundant than aquabilis males. So there's stronger selection, especially on this rare female, to avoid attention and courtship from heterospecific males. So as one last thing that I wanted to point out, I think this becomes additionally interesting because of shifting species ranges that I noticed in where we were collecting the damselflies and who we were finding where, and then Waage's 1979 report on where these sympatric populations are. So Waage showed sympatric populations in this range of blue, and our surveys indicate that this is the sympatric range now. So, so about 40 years later, this southern boundary of Caloptrix aquabilis has moved north about 300 miles. Who knows why exactly, but perhaps for the reasons that other insects and ectothermic organisms are showing also a northward movement toward the poles because of stream isotherm shifts due to climate change. So perhaps that's the cause here. But what this means is that we can now study all sorts of different populations and their mating preference in terms of sexual selection and species recognition, even in recently allopatric populations. So for example, populations here in the middle of Michigan, which used to be sympatric not all that long ago, do those males still show, do they harbor some preference for their own relative to the other species because of this historic selection pressure? So the preliminary data indicate, I think, that sexual dimorphism in these species may result from selection that differs in male and females, different selection pressures, relative selection pressure of sexual selection versus species recognition, and that there's some evidence that that this theoretical solution to conflicting selection pressures may be resolved by sexually dimorphic traits. So I've already acknowledged many students who have helped in this, who have done a tremendous amount of exploration and creative problem solving. And many thanks also to my collaborator, Jackie Brown. Here's a painting I made of him catching these damselflies in Ontario. And my institution, James Madison, and Michigan State for also their support. And thank you all for listening.
Thank you very much, Idelle. Very lovely talk. If anyone wants to talk to her directly, yes, please do so. So there you have a question. Can you read it?
Oh, I see it. Thank you, Mariella. They wanted to ask what might explain the asymmetry in displacement of female choice in the sympatric zone. Yes, which is only really observed in one species, the aquabilis. Could there be more cost for these females to make a mistake? I think that's exactly what's happening. I think there's strong selection on these females in particular. So the aquabilis females are the rare ones. And so that means the heterospecific male maculata are the common ones. So if you view it from selection on the male perspective, it's not a big problem if they mate with the wrong female. It's just some lost sperm if it's the heterospecific female. But for females, it's a much greater cost because they have fewer eggs. So wasting a whole bunch of eggs with the heterospecific sperm is a bigger problem. And for the rare female species, probably it's more likely, just by chance, to happen more to them than the common species of those females, because they're more likely to be approached by conspecific males.
Right, thank you. I'm going to take this occasion to ask you a couple of questions, if you don't mind. You have some more questions. Okay. Please go ahead. Yeah.
Mariella says, are there any studies of whether females may expel more frequently or have some kind of heterospecific sperm recognition? No idea. There's some fascinating things that we could potentially do because these females have these sperm storage organs. What are they doing with them? Nobody knows. There's also a recent study which indicates in the maculata that the males have these two processes to scrape the sperm out of the storage organs, but they preferentially have like one side that's bigger. They call it penis handedness. So a left scraping or a right scraping, and that they can't maximize both. So maybe there's some fluctuating selection on females and some sexual conflict. Who knows, but it would be great to study. And Ulysses says, my question is there any relation between male pigmentation and thermoregulation? There have been some studies on some species that have a dark wing pigmentation in Japan that indicate that the males with the darker wings are more active, and there's some selection on the males with darker wings because they're more active, but not in the species that hasn't been studied.
Yeah, related to that question, Ulysses' question. Yeah, I mean, you showed us this latitudinal potential displacement in which the dark species is sort of is losing some of its distribution range, right? So I suppose that can also occur altitudinally, right?
Yeah.
In the same fashion, right? I mean, it would be nice to use the same data that you have, link it to the altitude, right? To see whether that is what is also happening.
Yeah, I think that would be great, because I think that's clearly the alternative explanation for this kind of latitudinal pattern.
And there is, there is evidence that dark insects are doing poorly in this climate change processes. And although you have a difference in how many years, like 40 years difference between your data and Waage's data. Yeah. I mean, that can be like a case how, and it can be also nice to see like how this conflicting selection is driving one species to extinction, possibly locally at least, right?
Yeah, exactly. I'm curious whether it's or whether it's both selection on adult pigmentation or larval habitat, and whether the stream changing temperature might be affecting where the adults are displaying. Who knows?
So you have some more questions over there, Oscar and Chris Betty.
Okay, Oscar asked about the male terminal appendages, which is a fascinating question because in many damselfly species, the male terminal appendages are specific shapes that differ between species, and they match conspecific female plates in their females' necks when they begin copulation. These Colopterigidae are interesting, and they kind of jumped off that. They jumped ship in that lock and key mechanism, in that the male terminal appendages are so big it goes around the female neck entirely and kind of bypasses that species recognition system. So I don't know that it's been looked at a lot, but they probably, I would predict that they don't vary in form, but we should look into that more. And then Chris, hi Chris, notes, thank you. Are there any differences in daytime activity levels between the two species, especially in sympatry? I don't know. We have some preliminary data, but we don't know yet. That's a good question, which would address somewhat Alex's point that maybe there's some temperature thermoregulation effects too.
Yeah, clever, very clever experiments though. I've really loved your the powder marking, the sort of a cutting of the wings and replace them with the other species wings. That was, I mean, I tried that for years, you know, and now I'm surprised and I'm fascinated that somebody else finally did it. You know, this is so cool. I really liked it. I really also liked the idea that the zigzag flight may be related to avoiding predation, and with that I would say, yeah, my prediction would be that yes, in those places where predators such as birds are more common, yeah, you're gonna expect that odonates there in general are gonna use more of these erratic flights, right?
Yeah. So it's certainly, I think the first thing to do is to study in this system the sexual selection and species recognition, but clearly there are other selection pressures that are happening at the same time, including predation. And does that conflict also with sexual selection in the way that Svensson studies indicate in Sweden does?
Yeah, very nice. Can you? Is there any way that you can share the brand of that field scanner that you use? Because I'd love to have one. Because it's just amazing, right?
I've been waiting for years to get a portable one that's battery powered. But
right. Yes.
And they're really small, so they're great.
Yeah. The link. And then you connect it to your computer over there in the field as well, or no, how does it work?
By itself with the battery, and it has a little data card, like in a camera, like a little SD card, and then you can just take the card out and download the scanned images later.
There you have another question by Mariela.
Could the dark coloration of females be associated with some other female trait, perhaps their condition, size, or weight? That's a great question. I think we can answer that question when we have a big enough sample size of females in which we have their bodies, but then we change the wings. So I think that's the power of being able to manipulate the wings specifically to say whether there's other things that are going on. But I think first we can just have so many scanned images, thousands of females and males. I can just see it first if there's a correlation between these traits. And then we can disconnect that correlation with the wing surgeries to see selection on the different traits.
Let me say this first. There is another interesting species that overlaps the distribution with these calopterics, which is Tethered in Texas. You know, that is a dark species too.
Yeah, I'm actually in Texas right now.
Oh, right. Oh, you can go out then.
Not normally in Texas, but yeah, just nearby, there's Tishia, and they're in the same flying zone as Heterina americana.
Right. And you have aquabilis there, or
there is maculata, but not
maculata. Yeah.
So I think there's a lot of potential. I described just these two species that I've been studying in Michigan and Ontario, but I think we could potentially do the same sorts of studies in Texas, Arizona, and in Mexico, because I think the same things are happening with Tishia and Heterina.
Right. So Oscar has another question again.
Is there variation on mating success throughout the mating season? That's a great question. At the beginning of the season, in other species, there seems to be more females than males. I don't know whether the sex ratio varies over the season. I imagine it does. But what I've noticed so far in the last field season, when we were doing those tethered experiments, we started doing them in the middle of the season in Ontario. And the males, like I haven't summarized this data to show you, but the males preferred the dark winged females, like they always see, even when they're given the light winged option. But then we did it again at the very end of the season. And there weren't all that many damselflies, females flying around anymore. And then males appeared to not care. They were also mating with the light winged ones. They were mating with everyone. So I don't know if there's something about like when there's a time limitation to the end of the season that it's costly to pass up mating opportunities more so than make mistakes or conspecific, heterospecific mating potentially. So I think mate choice can change over the season, and it's something we should pay attention to.
I was going to say, again, with the Tishia species, Tishia males do not court females. That's interesting because they just go and grab them, you know. So it's a different thing. So that's why it may be like, go ahead, sorry.
Well, there might be then some interesting predictions about what sexual selection would be. It wouldn't be from females on males, it may be males competing with each other. And then species recognition of males grabbing the females.
Yeah. So you have another question, yeah.
Oscar asked, when I changed the wings on the males, do they change their wing mating displays? So far, while you can change the wings on males, we've really just been doing it on females who don't do the wing displays. I mean, they display their wings and that their wings are visible. But the males who have more elaborate wing displays, we haven't tried doing the little wing surgeries on them. I think that would be useful to see, though.
One other question is, you think there is a role for memory there? I mean, that males can like memorize females and learn eventually?
I think there is. That's another complicating factor that I'm having trouble working out. How do you make a study that also addresses potential memory or learning?
Yeah, because I remember, you know, those old papers by Waage and John Alcock that, maculata, I think it was that males were able to see which females they were guarding, right? So when they were guarding females, when females were ovipositing, males knew which ones they have already mated with, so they didn't mate again with these females. Yes, I think it. Yeah, so they that was like the idea of that they learned, right, that they at least could recognize at least in a short term, in a short time window, that they knew which species was that apparently, right? So it would be fascinating, and actually that was one of the topics that Greg Grether was talking a lot about in one of the papers that he published on. It was like a review paper, that there is a role of memory here, that and apparently memory may, because that result of maculata with that John Alcock produced years ago, with males learning or males being able to distinguish which females they just mated with, that ability doesn't seem to be common, or at least there is variation, interspecific variation. Not all species have that. Some species, for example, the ruby spots, males repeatedly can mate with the same females that they have just mated with. They can do that. They don't apparently know. They can't see the difference apparently. So that's why it's interesting. And with Calopteryx is a different story, right? I mean, there may be more pressure to avoid, I don't know, superfluous matings. I don't know. But it's again, it's an interesting issue here. I mean, whether they can learn or not.
Yeah, I think I have to put a lot more thought into how to study that. But I think it's a major question.
Mm-hmm. Yeah. So, guys, if you have any question, even in Spanish, I can translate that. So don't worry about that. And did you type your email address?
I did, but it's at the beginning. Let me just type it again.
Oh yeah, it's over there. Idelle Cooper at gmail.com.
Mm-hmm.
Yeah. One of the things that we do usually is that we ask our speakers whether they would be fine if we upload their presentations on our channel, our lab's YouTube channel. We can send you the link for that, so you can also show it to your students if you want.
Yeah, that would be great.
Well, you make me. You gave us some inspiration again about going back and doing some work with them, with these lovely animals, especially the genitalia and so much. I was thinking about like whether there were some predictions in relation to allopatry, sympatric, differences in genitalia, and because we used to do some of that work in the past in Europe with the different Calopteryx species they have there, and there was some variation, actually, some geographical variation, yes, in genitalia. So I was wondering if you have the samples. I mean, you can send us the samples, and we can just dissect them and measure them. They are very rigid, you know. These are rigid structures, so they're not gonna. They're fine. I suppose you have them in alcohol or something. Ethanol.
Alcohol, but I have most of them just in the freezer in envelopes.
Mm-hmm. Yeah, just if you can send us some samples, at least, I don't know, like if you can sort of subsample your samples, like sending us like those in the north from maculata, those in the sympatric region, and those in the south range of Aquabilis, and then we can just check them and spot with. That would be nice. It would be nice to see whether there is some sort of a character displacement at the level of genitalia.
Yeah, I just assumed there wasn't, but we gotta look.
Yeah. I mean, there is hybrids, right? Did you ever see any hybrids?
Well, I saw some that might have been, but I didn't do any genetic studies. But Mullins did a genetic study, and found some introgression, a low, but it exists.
Interesting. Anyway, Idelle, I think that's it. Yeah, David Schneider has just sent you the link of your talk, yeah, on YouTube. So you can check it from there. And thank you very much. It was a lovely talk, very, very inspiring indeed.
Thank you. It was so nice to talk about it and to hear your responses, and I look forward to talking more, obviously.
Definitely, yeah. Okay, have a great day, and we'll see you for other reasons at some point, right?
Yeah, exactly. Thanks so much, everyone.
Thank you. Bye, everyone. See you in 2 weeks.